Hello everyone, welcome back to another session of RNA biology, and this was the last slide where we stopped in the previous class, and this is a cartoon of the ribosomal RNA export through the nuclear pore complex, and we discussed in detail about the way in which the ribosomal RNA and the ribosomal RNP in fact pass through this nuclear pore complex with the help of Fg containing protein which is lining the lumen of the nuclear pore complex. And those who are interested can read this review which is published in Nature Reviews in Molecular Biology a few years ago, which is a quite exclusive review which has also linked to several latest publications in the field. Now let us see what are the major disorders or diseases that is linked to the RNA movement or to and from the nucleus. So what are the possible defects and troubles? We also saw some splicing defects and the diseases related to that. So defects in the mRNA nuclear export pathway can have direct consequences on human health and development. Health means day-to-day life, development means embryonic development. It can even cause a defective embryonic development, etc. So one has to be, or one system or one organism system has to be perfect in order to have a normal development and normal way of living. Let us see some example. Hijacking the endogenous mRNA export machinery by viruses can often have a deleterious effect. You may realize if you have an infection, say virus infection, of course your body responds. Normally T cell mediated response comes into picture, which takes around 6 days to reach to its perfection. So when you have a viral infection, let it be any infection, whether it is a chicken pox, whether it is COVID or any other, doctors normally say viral infection or viral fever, etc. Before you get that real fever is your body's response, right? The high temperature, etc. You will have 1 or 2 days of uneasiness, that malice. You will feel not have feeling to have food, like you have, some people have body ache, or not. We normally say that I don't have good mood, I'm not in good mood. So this mainly because the virus which entered your body is hijacking the machinery. What you have, it is entering, it is increasing in number, because initially our immune system may not be capable enough, or surveilling the immune system may not be powerful enough to detect the single virus or 2 virus, etc. Although ideally it should detect, but it can take time. So in this meanwhile, the virus will enter the cells and hijack your machinery for its own benefit or its own utilization or propagation or survival. So this can have too much low energy production in your body. The ATP production, other normal metabolic reactions will be compromised, and that is the reason for that, the so-called feeling of not so good feeling. And before you get the onset of fever comes when your body detected there is some problem and body tries to kill it by elevating the body temperature. So this is one situation where the viruses, when they hijack the transport machinery, and of course virus will hijack pretty much every machinery in your cells, but transport machinery also virus will hijack, which can give lot of trouble. And another example, fragile X mental retardation protein called FMRP, and which interacts with a homologue of NXF1 gene, that is called NXF2, and destabilize it. And this kind of situation, the FMRP is important in the movement of RNA across the nucleus. We have seen it, some examples earlier. So this FMRP may not be effectively available for the transportation function. So that is another example. Another example is the disease called osteogenesis imperfecta. So osteogenesis imperfecta is characterized by fragile bones. So normally the density of the bone becomes lesser and lesser, which makes you vulnerable for bone or fracture, bone fracture, etc. And this osteogenesis imperfecta is associated with aberrant mRNA export. So here a specific splice site mutation in one of the cohort of patients has been reported to result in defective splicing and nuclear retention of collagen mRNA. We know collagen, mainly different types of collagens are there, mainly 4 collagens are important, collagen 1, 2, 3 and 4. Although the family of genes are around 20 different genes are there approximately. So collagen is important for maintaining the flexibility of the tissue. It is there in wherever you have tissues, that is we so called the structural cells, which we call they don't have any specialized function, those cells, like fibroblast cells, which is kind of covering the gap or filling the gap between the differentiated tissues. Say when you have a wound in your hand or leg, so the gap is filled by fibroblast. So many times your collagen is there pretty much everywhere. It basically makes sure the smooth transition, smooth movement of cells among themselves, and also the your ear pinna, nose. So these are all very flexible, you know that. This is mainly because we have got cartilaginous tissue, so which is calcium derived, but it's not very strong, it is not very dense as bone. Your bone won't bend like this, right? So like that, many places we have tip of your nose here, behind here it is solid, but little bit down it moves, your nose, there will be slight flexibility is there, your ear moves around. So there here and there some places you have got cartilaginous tissue. Again, the credit goes to the collagen, the distribution of collagen, like you are able to move your hands, like some part of your body you are able to turn and twist, etc. The credit goes to the collagen. If you don't have adequate amount of collagen, your bone formation, the maintenance of your bony structure, etc., everything can get compromised. Remember, your bone keep decalcifying and continue to calcify. So it doesn't happen like in around 7 years, approximately 7 years, your entire skeleton is replaced. So if a person is 21 years old, he is on the 3rd version of his skeleton. So you can keep multiplying. If a person is 70 years old, he is on the 7th version, 10th version of his skeleton. So your skeleton on an average stays only around 7 years. So it doesn't happen on one day morning the entire skeleton is replaced. It continues to replace. So why I am saying is, the bone, you may have a dense bone right now, but that doesn't mean that it will continue like that forever. So the food you take, the rate at which the bone density is maintained, or the rate at which the calcium deposition happens in your bony parts, decides how strong the bone you have, etc. So one such disease that comes, osteogenesis imperfecta, is due to a splicing defect, and the nuclear retention of the collagen mRNA happens. Because if there is a splicing defect, collagen doesn't come to the cytoplasm, collagen RNA doesn't come to the cytoplasm, because it is retained because of a splicing defect. So lot of patients it is reported that the collagen RNA is not coming to the cytoplasm, hence the collagen protein is not being formed effectively, and collagen is secreted out of the cell and it functions outside the cell. Like I told you, it acts like a glue. Lot of collagen and their mutation are very important. Like if certain mutations or even isoforms of collagen allow people body very flexible, some people body will be very flexible compared to the other. So collagen, being a structural protein, can tolerate to some extent some mutations, because it's not as important as a functional protein. I'm not saying that mutations in structural protein are unimportant, not like that. They are also important to maintain their amino acid sequence, but sometimes some structural proteins tolerate variations, and some such changes from the general population allow much flexible body. Body is very flexible, you can bend it, you can twist it, whatever way you want you can do it. The credit goes to the collagen deposit between the joints. So bone, you cannot bend it, whoever it is, the bone cannot bend it, but still the bone will remain tough. Credit goes to the nature of the collagen. So if collagen is not being formed adequately, then you can end up getting situation like osteogenesis imperfecta, where you are more vulnerable for bone defects or bone damage or bone breakage, etc. Those who are interested can read some of these references. Exporting RNA from nucleus to cytoplasm, which is a Nature review, Molecular Cell Biology, came few years ago, and mRNA nuclear export at a glance. This is another review which is published in Journal of Cell Science in 2009. So those who are interested can read it, because it will make your impression about the subject much better. Now we will move on to another topic, mRNA decay. We know that any mRNA that is formed from the cell has to have a lifespan. You do not want an RNA to stay forever. No matter how precious the RNA is, no matter how tough to make this RNA, like we have seen dystrophin, it goes in megabase size, and that produces around 17 kb size RNA, and so many exons are there, so that splicing, energy consumption, everything is too much is there. That doesn't mean that an RNA should stay forever. That can give lot of problem. Say for example, you think about an RNA is important during the development of an organism, and once that development is done, you don't want it. Say you go to a restaurant and have food, will you carry the plate with you? No. Will you carry your hand, just like, I love this food, so in between I can, have a taste of it. So will you carry? Normally no. However tasty, however great the ambience and place, you will wash your hand and you will come out as you entered inside. Of course with belly full, but that is not obvious. You went for that purpose. Same way, when an RNA is being formed, it has to perform the task of it. Say whether it is a structural RNA or an mRNA which is supposed to produce proteins. Once the job is done, the RNA should be cleared. Otherwise what will happen? It will continue to do the same job, and that will be overdoing it. And say a proliferation, an RNA or a protein which is facilitating cell proliferation. Cell proliferation means multiplication. Say you got a wound, your hand got cut with a blade and there is an open wound. Now after maybe a 10 days or 1 week, that wound will be closed. And who closed it? By cell proliferation. New cells are formed, they are filling the gap. Just like your house wall is broken, you cannot cover it with hanging a calendar or putting an almirah in front of that. You have to bring bricks, cement and seal it, and so that the wall is restored back. Our body also does the same thing. Produce cells, fill the gap and make the damaged part look normal. Now if the genes responsible for the cell proliferation or the multiplication of cell is not stopping once the job is done, it is still on. So cells are continuing to divide. Now it's dividing and dividing and dividing. So what the situation is called? It is called cancer. It is a problematic situation. You don't want that. So why a normal wound, when you heal it, you don't end up getting cancer? Because the RNA or the gene regulation are stringently controlled. So they are controlled in two ways. One way is stopping the production of that RNA, one way of regulation. What is the other way of regulation? Degrade the existing RNA. So if it stays back, it can give rise to lots of trouble, lots of problem. Like you may have seen in many situations, like if there is a communication between some high level officials, you don't want, you can be tracked by enemy countries. So what they do, if it's a prime minister or a president making a communication to a high level official, they immediately damage that phone contact, whether it's a mobile phone or whatever, they just to make that particular call. Then that card or connection, everything is disrupted. If they want to make one more call, they call with a new number. They call it, communicate it, and again they disrupt it, because they don't want their positions to be revealed to anyone. So they immediately, if it stays back, it is possible that it can give rise to lot of troubles. So they don't want that. RNA also like that. When you have a job is done, you have to degrade it. So we have to understand that RNA's degradation is much more important than even its production. If it is not degraded in time effectively, then that can lead to lots and lots of problems. So this has to be stringent also. You don't want like morning 10 o'clock in a cell it turned on, okay, RNA degradation pathway, let it be on, and all the RNA in the cell is now damaged. That way it cannot happen. It has to happen to those, if there are in a cell there are 100 different types of mRNA are there in a given time, you don't want all the 100 to be degraded, you want just 10 to be degraded. So remaining 90 should stay back. So RNA destabilization is a vast topic. Lots of people dedicate their whole career in understanding the RNA destabilization, RNA's degradation. It is not an easy topic. RNA, we all know that it's a highly vulnerable molecule. To degrade RNA, you don't need to do anything. It's like your sweat, your tears, your saliva, everything has got RNAs. So RNA is basically fighting against its degradation. However, when it comes to selective degradation, if you fail to do so, then there can be lots of trouble to the cell, including developing complicated conditions such as cancer. So RNA degradation or RNA decay is an important phase or important science of RNA biology or important part of RNA biology. So RNA usually starts its quality control right from its production. We all know in any company when they are producing a product, if there is any damage, then they simply will not proceed to the next stage. Because if so, first of all, company's reputation will go. Second, that product may not be optimally usable. So any defect, if it is there, you will simply discard it. Some of you may have used many centrifuge machines, right? Like some class of centrifuges are called ultra centrifuge, which can run RPM of 1,30,000, 1,50,000, those kind of RPM. So the machine will cost, say if it is costing around 1 crores of rupee, the major part of that goes into the rotor, because rotor is not even the motor. You may wonder, oh, motor you can make it however powerful you want, you can, even people make it, aeroplane also you make the motor, but the rotor which you are making has to be the most sensitive part in a centrifuge, especially in an ultra centrifuge. So it is made of special alloy, and they have to cast it multiple times and screening for the defect. So it is said that if you make around 12 rotors of the same dimension, same size, only one can be marketed. The remaining 11 has to be discarded because they will not pass the quality control. That is why the rotors are one of the costliest of the ultra centrifuge part, where they can go in lakhs, like 12 lakh, 50 lakh, 20 lakh, like that. It goes lot of, and when you look, you will say, oh, it's a metallic part made of some alloy, how does that matter? No, it is discarded. Lot of them are discarded. Same way, RNA is maintained very stringently by quality control at every step. So we can ask the question, why do cells need to control RNA quality? And I don't think I need to discuss this, because by now you know how important the RNA is, how important a given sequence of RNA is. Only a small portion of RNA is repaired. If an RNA has got a defect, if it is not passing the quality control, it can be repaired to some extent by using RNA modifying enzymes, etc., etc. But RNA editing also can be called as a kind of repair, because a given RNA is problematic, it is not going to be useful, so hence it has to be repaired, it has to be fixed. So only a small portion of RNA is repaired. That means any RNA that do not pass the quality control must be marked for degradation. So it starts from its production, that is right from the nucleus. Now let us see RNA quality control. What are the steps? What are the stages it happen? Transcription, QC stands for quality control, and it can cause degradation, or it can be marked for degradation right from the transcription. Capping, if capping has an issue, 7-methyl guanosine cap, if any problem, cap doesn't work effectively, it has to be marked for degradation. Splicing, if there is any defect, should be marked for degradation. Polyadenylation, if there is any problem with the polyadenylation due to whatsoever reason, it should be marked for degradation. Export to the cytoplasm, an RNA, it is capped, spliced, polyadenylated, but somehow it is not effectively forming the RNP. Hence it is failing to get exported, it is lingering, standing here, should be marked for degradation. And translation, an RNA which is translating in the cytoplasm, mRNA translating in the cytoplasm, sometimes it does one round properly, then it detects something is not right, the protein that is formed is not right, it will be marked for degradation. So every stage it has to be marked for quality control mediated degradation. Then comes if it is utilized properly, RNA, it was perfect, passed quality control in every stage and it did its job. Still that doesn't mean that that RNA is eternal. That also should be marked for degradation, because after some rounds of say protein translation or maybe some rounds of its actual function, the RNA lose its integrity in terms of its secondary structure and ability to form RNP. It was a perfect RNA, just like you can see your mobile phone. It was perfect when you purchased. After a few years, why it goes slow? Although you did not do anything, because the parts inside, the electronic parts inside are not able to conduct electricity or electrons as it was used to do it. It is still working, but in a very slow and defective manner. Any instrument for that, including you may not have that much energy as you used to have when you were 5 years or 6 years old. That time you had full of energy. As you grow old, you are energetic, you are muscular, but it will slowly come down. Same logic applies to RNA also. Although it doesn't have any defect, it is used a lot, then also it should be marked for degradation. So RNA decay, in most studies of RNA decay are done with mRNA with a purpose, because mRNAs are diverse and mRNAs are unique, because a given cell, a given mRNA may have a longer lifespan, but that may not be so for tRNA, ribosomal RNA, etc., because they are mundane RNAs, like they don't have any they don't have any special identity to them, although they are important, although they are essential, but they have a mundane job or routine job. Whereas mRNA, that is not the case. A given mRNA expressed in a cell can completely transform the outfit of that cell. Means a kidney cell you can convert into liver purely by expressing the mRNA. You may have heard about Yamanaka factor. Yamanaka, who converted the fibroblast cell, like we were discussing earlier today, fibroblast cell into stem cell. So just express few RNA, few genes you express, and he expressed just 4 genes. 4 genes you express, we call them as Yamanaka factors, and we also call them pluripotency factors. Just express 4 of them, 4 RNA. Fibroblast, which was doing no special job, now became stem cell, where you can convert them into any tissue. Stem cells can be converted into any tissue in your body. So they became pluripotent stem cell. So RNA as small as 4 can convert huge change in the outfit of the cell. That is why a lot of decay studies have done in mRNA. Cells need to remove mRNA that damaged or do not need anymore. So don't think when an RNA is degraded, it is a damaged. No, it is no more needed anymore. Just like I gave the example of, once a communication is done between 2 high power officials, they damage the phone or break the phone because they don't want any tracking. Same way. So mRNA decay pathways are found in both nucleus and cytoplasm, because both places there is RNA, and quality control happens both in nucleus and cytoplasm based on which stage of RNA production is going on. So mRNAs have 2 stability determinants, that is 5 prime, that is the 7-methylguanosine, and at 3 prime the poly A tail. So these are all the 2 boundary yardsticks. So if RNA lacking any of these should be marked for degradation, because both are needed. And the 5 prime cap binds to ELF4E, which we saw that CBP will be removed as it comes into the cytoplasm from the nucleus. The mRNAs, the 7-methyl cap bound CBP will be removed and ELF4E binds onto it. And the poly A tail binds to the poly A binding protein, PABP. So poly A binding protein is very important to have identity to the poly A tail of an RNA. So ELF4E binds to the cap, in this I am talking about in the cytoplasm, and PABP, poly A binding protein, bind to the poly A tail. And decay can start only if one of the two structures are removed or there is a endonucleolytic attack. So if ELF4 is bound down to the 5' end and PABP bind down to the 3' end, everything is fine with the RNA, but they are still vulnerable for endonucleolytic attack, where you are getting cut by an enzyme right in the middle of the RNA, which exposes two tails. One 5' end is exposed, a 3' end is exposed, new because of the cut. That can be utilized by exonucleases and can cause degradation. So it has to fight against the endonucleolytic cleavage by enzymes also. So we will see more in detail about the RNA decay pathway in the next class. Thank you.